Welcome to this presentation on affinity tag strategies and magnetic particle purification of recombinant proteins. In today's session, we will explore various affinity tag technologies used in recombinant protein purification and highlight how magnetic particles, especially those developed by CD Bioparticles, offer practical and efficient solutions. Let's start by exploring why affinity tags are so commonly used in recombinant protein purification. Affinity tags, such as His tags, flag tags, GST, and strep tags, are short peptide sequences genetically fused to the target protein. Their primary purpose is to make purification more efficient, selective, and reproducible. These tags bind specifically to complementary ligands, which are immobilized on a solid support, such as magnetic beads. This highly specific interaction allows researchers to isolate the target protein directly from complex biological mixtures like cell lysates, with minimal background and high purity. This approach not only simplifies the workflow, but also preserves the biological activity of the protein, which is critical for downstream applications in structural biology, drug discovery, or therapeutic development. The most commonly used tag is the His tag, which utilizes immobilized metal affinity chromatography. Histidine residues coordinate with metal ions like nickel 2+ or cobalt 2+, immobilized on a solid matrix. The His-tagged protein binds to these metal ions and is then eluted using imidazole. This method is efficient and well-suited for magnetic bead-based purification. Other popular tags include flag and strep tags. Flag tags are recognized by anti-flag antibodies, offering high specificity, while strep tags interact with streptactin under mild, physiological conditions. These systems are ideal for purifying proteins that require native conformation. CD Bioparticles provides both flag tag and strep tag compatible magnetic particles. Traditionally, affinity purification has relied on packed columns combined with gravity flow or sophisticated systems like FPLC. These methods, while effective, often require bulky equipment, trained personnel, and time-consuming steps such as centrifugation, buffer exchange, and column regeneration. For many laboratories, especially those with limited space or resources, this can be a significant bottleneck. Magnetic particle-based purification is revolutionizing this process. Instead of relying on packed columns, magnetic beads offer a faster, simpler alternative. The magnetic particles are functionalized with ligands that capture target proteins from a lysate. Once binding is complete, a magnet is used to quickly pull the beads and the bound proteins out of solution. No centrifugation or vacuum filtration is needed. This column-free workflow dramatically reduces hands-on time and simplifies the process. It also scales easily from microliter volumes in 96 well plates to milliliter or even liter-scale preparations in tubes or flasks. This is especially useful for labs with limited infrastructure or high-throughput demands. Magnetic particles, often composed of iron oxide cores coated with polymers or agarose, offer superparamagnetic properties. They can be functionalized with various ligands like nickel nitrilotriacetic acid, antibodies, or streptactin. Their small size and large surface area enable fast and efficient protein capture, making them ideal for both small-scale and automated applications. Let's go through the main steps of purifying recombinant proteins using magnetic beads. We start with cell lysis, where cells are broken open using a buffer that preserves protein structure and function. Next, the lysate is incubated with functionalized magnetic beads. These beads are coated with specific ligands or antibodies that bind to the target protein during incubation. A magnetic field is then applied, quickly pulling the bead-bound proteins to one side, allowing unbound materials to be removed easily. Washing follows to eliminate nonspecific binders and contaminants, improving the purity of the sample. Finally, the target protein is eluted by adding a buffer that breaks the bead-protein interaction, yielding a purified protein solution ready for use. This workflow is efficient and preserves protein functionality. At CD Bioparticles, we offer a range of magnetic beads tailored for recombinant protein purification. Our absolute mag nickel-charged particles are perfect for His-tagged proteins, while anti-flag and strep-tag beads serve other purification strategies. All our products are optimized for high binding capacity and purity. Let's look at a case study using Ni-charged magnetic beads. In this experiment, His-tagged proteins from insect cell lysates were efficiently purified using magnetic particles. The result was over 50% recovery and more than 75% impurity reduction, demonstrating the practical effectiveness of this method. Beyond his tags, CD Bioparticles provides magnetic beads compatible with FLAG, STREP, GST, and Protein A/G systems. This versatility supports diverse applications, from antibody purification to complex pull-down assays. Whether your target protein is cytoplasmic or secreted, we have a solution. Recent advancements have improved magnetic bead performance through core shell nanostructures and surface optimization. Many beads are also reusable, making them cost-effective. Magnetic particles are also being integrated into hybrid and automated systems for scalable protein purification. For optimal results, select bead size based on your workflow, micron scale for manual use and nanoscale for automated systems. Optimize buffer composition, binding times, and elution conditions. Our technical support can assist with detailed protocols tailored to your protein. One of the major advantages of magnetic particle-based purification is its exceptional scalability. This technique can be easily adapted from small-scale manual procedures to large-scale automated workflows without compromising performance. Thanks to their compatibility with standard 96 well plates and robotic liquid handling systems, magnetic beads are ideal for high-throughput screening and purification tasks. This makes them a perfect fit for modern laboratories looking to increase efficiency and reproducibility. CD Bioparticles offers a wide range of magnetic beads specifically engineered for automation. Our beads demonstrate consistent performance across platforms and are well suited for both research and development as well as large-scale manufacturing environments. Whether you're optimizing a protocol in the lab or streamlining a production pipeline, our magnetic bead solutions offer flexibility, reliability, and ease of integration. To summarize, Affinity tags streamline recombinant protein purification, and magnetic beads offer a fast, reliable, and scalable approach. CD Bioparticles delivers high-quality magnetic products that support various affinity tag systems. Contact us today to request samples, protocols, or technical collaboration. Thank you for your attention.